So the blueprint of life is actually DNA. So DNA is going to be used to synthesize a sister molecule, which is going to be RNA, and then the RNA is going to be used to produce and synthesize a protein. So this flow of information from DNA to RNA to protein is what we refer to as central dogma. And yes, it applies to all forms of life, meaning it doesn't matter if you're bacteria or fungi or a plant or a little tiny fly on a wall or a human being, you will have that process happen in your cells. So let's quickly compare DNA with RNA. The nuclear bases are going to be different, and I'm going to point out one major difference here. DNA has thymine and RNA has uracil instead of thymine. And if you notice right here, the difference is going to be the absence of this methyl group right here. And then, of course, the sugar molecule is going to be different between DNA and RNA. Sugar in DNA is deoxyribose, and RNA is going to have ribose. Deoxyribose, notice right here at the 2' end of the sugar molecule, is missing oxygen. So we're going to say it's deoxygenated. So this is why we refer to DNA as deoxyribonucleic acid. And then the RNA molecule has that oxygen at this position, at the 2' end of the sugar. So RNA comes in all sorts of forms, actually. Most of it is going to be single-stranded, like your mRNA, the one that you use to produce a protein. But then we also have a list of other RNAs that will have to carry out very different specific functions. So an example here would be tRNA. This participates in protein synthesis, and you can see how it's actually, it would be like a string at first when you synthesize it, but then it's actually folded into loops. And then this rRNA is found in a ribosome, so this is ribosomal RNA, and you can see lots of loops and bends and pins that are formed there. So it's actually very complex. So please do not think that RNA is always single-stranded. That would not be the case. So this is where we perform that first step in gene expression, where we go from DNA to RNA. So we're making this sister molecule, and we're not going to make a copy of the entire DNA molecule. We only need to select a specific gene. I'm going to assume this is the gene that we want to express, and you can see that the DNA has been separated so you have 2 strands that are separate. So one of these strands is going to serve as a template. And this template is what we're going to use to synthesize the RNA molecule. We're going to apply base pairing rules. So if DNA had an A in its nucleotide, so this is the adenine, is going to pair up with uracil in RNA. And if it had a C, it's going to pair up with guanine. So you can see that the main enzyme that's going to synthesize the RNA molecule is going to be RNA polymerase II. So it reads the template and allows for the addition of nucleotides based on the order that the template dictates. So you can see the next nucleotide is going to be an A because I had a T right here, so we would add it A. And we continue to break these hydrogen bonds and read the gene until we actually run into a termination sequence, which basically means the end of the gene. And then we release the RNA transcript, and then we're ready to do other things. But in what direction do we actually synthesize the RNA? And the answer is from 5' end to 3 in this direction, because we can only add the incoming nucleotide at the 3' end. Because as I said before, the 3' end of that sugar molecule in a nucleotide offers the hydroxyl group, and that's where the reaction is going to take place to link up those nucleotides. During RNA elongation, RNA polymerase builds the RNA strand using ribonucleoside triphosphates, or rNTPs. The key reaction happens when the 3' end hydroxyl group of the growing RNA strand attacks the alpha phosphate of an incoming NTP. So this forms a phosphodiester bond and extends the RNA chain. When that bond forms, pyrophosphate is released. And beta and gamma, that's your pyrophosphate, is then hydrolyzed into two inorganic phosphates. And that hydrolysis provides the energy that drives the reaction forward. So just like DNA replication, except that we do not need any primers to get the process going, remember, RNA synthesis is powered by the cleavage of high-energy phosphate bonds in the NTP. It's not by ATP separately and not simply by dehydration. So transcription can be summarized in the following steps. We can say initiation happens first and then followed by elongation and termination. So if we look at the gene we want to express, we're going to see that there is this regulatory sequence that's sitting within the DNA molecule. And this regulatory sequence is basically a promoter, acts like a molecular switch that can turn on the gene expression. And you can see this gene is right here. And then finally, there's a sequence to terminate transcription. So when RNA polymerase is recruited by transcription factors to the transcription site, RNA polymerase is going to synthesize the RNA in the direction of 5 to 3. Remember, I already mentioned that. And you can see the elongation is happening here. The 5' end is right here, and we're adding the nucleotides, the incoming ones, to that chain. And then RNA polymerase runs into termination sequence and everything dissociates and transcription is completed. So these are the basic general steps of transcription. So now I want to talk about how do we actually initiate transcription. So if we look at the promoter region within the DNA sequence, the promoter is going to be, as I said before, that molecular switch that's going to allow us to express the gene. Promoter has a very unique sequence, and one of the sequences that we have here is called a Tata box, T-A-T-A. So the sequence appears to be conserved among many eukaryotic organisms. This Tata box is going to be that sequence that the first transcription factor is going to bind to initiate transcription. So now in this picture we see that we only have a few transcription factors because it's a super simplified drawing. So we have 1, 2, 3, 4, 5 here and then 6. But you know what? The reality is this process is so much more complex. We actually have about 80 and above the number of transcription factors. So they all come together and they build this huge protein machinery, and we're going to call that holoenzyme complex. And they recruit the RNA polymerase to the scene, basically telling RNA polymerase where the gene is located, which gene needs to be expressed. Without that, RNA polymerase 2 would not be able to express a particular gene because it doesn't have that specificity. So transcription factors are going to be the key proteins that are going to bind to the promoter region and other regulatory sequences, and they will recruit the RNA polymerase to the scene to begin the actual synthesis. And we can see during elongation process, and this happens in a 5 to 3 direction, we are adding the nucleotides one at a time, and we have this temporary RNA-DNA hybrid region that forms while it's happening. And then termination sequence is going to be somewhere right here, and then we say, okay, now we can actually terminate transcription. When transcription termination happens, obviously all the components are going to dissociate from the DNA. So I want to point out the general structure of eukaryotic gene. So we have the promoter region, and remember the promoter region is going to have that Tata box. And then we're going to have what we call 5' untranslated regions. So these units right here, these sequences of DNA are going to be important because they're going to be communicating with ribosomes when it's time for translation. And then we have exons. So these are going to be the coding parts in the DNA, coding for proteins. So they have those sequences that we need. And then introns are going to be parts that do not code for protein. And you can see we have exons interrupted by introns, and then the entire gene is going to be flanked by 3' untranslated region. And then upstream of the promoter, we have an enhancer. I haven't talked about that yet, but I will in a minute. So now the question is this: if we actually look at the DNA and we say, we know that only one strand of DNA is going to serve as that template. So remember, the template antisense means the same thing. But the question is, which one? How do we actually determine? So the quick answer to that, you have to look for the promoter. See where the promoter is located, and if it's given to you, there's your Tata box right here. That's also another clue. But first, what you want to do is notice the prime ends of the molecule, of the DNA molecule. So here's my DNA, 5' end here, 3' end here, 3' and 5. So now if I know that I have to synthesize the transcript in the direction of 5 to 3, I must start at the 5' end, which has to be complementary to the DNA strand. So is the top strand going to be complementary to my transcript or the bottom? What do you think? So you can see the top strand right here, we would have 5' end, and the bottom here is a 3' end. And I need to make a complement. You see, so clearly the bottom strand is going to be used as a template strand. So you can see if we had a T here in the DNA, now we have an A. If we had an A here, we have a U, and so on. And we build that transcript one nucleotide at a time until we run into that termination sequence. And then, of course, step 2 later is going to be translation, which you see right here. So now what I need you to do is this. Look at the 2 example problems. So this is the DNA molecule here. It's double-stranded, and you have the following RNA that was produced, and what you have to do is figure out which DNA strand was actually used as a template to synthesize the given RNA. So basically what you want to do is determine in which way RNA polymerase was moving to synthesize the strand, meaning how it was reading the DNA. Was it going from left to right? Was it going from right to left? What was the direction? You might want to pause this video here and give it a try. Okay, so I hope you gave it a try and you realized that the bottom strand here is going to be used as your template. So you can see this complement would be perfect because we got to grow that RNA in this direction. Therefore, the promoter region would be sitting on the left, and the Tata box would be sitting on the top strand. And then for the second scenario, notice the promoter would be on the right, and the promoter region, the Tata would be sitting on the bottom strand, and we would be going from right to left. So this is the direction of RNA polymerase, and RNA polymerase would be reading the top strand as a result. So we have 3' end, 5' end, G and C, C and G, and we keep going because we got to grow that strand in this direction. So here's a typical eukaryotic gene structure. You can see we have the promoter element, we have exons interrupted by introns, and we have termination region here. So upstream of the promoter, we will also find other control elements that are going to be involved in activating or transcribing that particular gene. We will notice that there is this what we call enhancer region, which is very important if you want to be able to increase transcription rate in a particular cell. Maybe you have housekeeping genes and they have to be transcribed on a regular basis. So these enhancers will come in particularly handy. So what happens here, when the signal is received by the cell to turn on a specific gene, the activator proteins are going to bind to the enhancer regions, and they are going to loop this DNA. And you can see the looping is happening right here. And then recruit other activator proteins, other transcription factors to the scene, and build this huge complex that is going to initiate transcription. Because at this point, RNA polymerase is going to be recruited to the scene, and this is how RNA polymerase will know that this is the beginning of the gene. This is what transcription needs to begin. If, for example, this enhancer is necessary to turn on the gene to begin with, and you have a mutation in that enhancer, you're not going to be able to express that gene. When we transcribe mRNA, before it can leave the nucleus, it has to be edited. So we call this process basically mRNA processing or editing. So 3 things are going to happen during this process. We will have 5' GTP cap added to the transcript. We will have a poly A tail added to the transcript. And then also we're going to be removing introns. Remember, this part was an intron and this part was an intron. And you can see your final mature mRNA molecule does not have any introns. So let's talk about each a little bit more. So addition of 5' GTP cap occurs shortly after the initiation of synthesis of mRNA. So it's basically co-transcriptional process. So the GTP cap is at the 5' end, and you can see it has 3 phosphate groups, and then this guanine is actually methylated here. The reason we need that is this cap is going to be necessary for translation so that ribosomes can attach to mRNA and carry out translation. So in other words, without this cap, translation is not going to occur. The addition of poly A tail is actually catalyzed by a separate enzyme, and the separate enzyme is called poly A polymerase. So it's using ATP. Those AMPs are going to be linked together in the form of poly A tail. So this long tail that's added to the mRNA is going to protect the mRNA from the enzymes, from the cytoplasmic enzymes that could potentially degrade this molecule. So, and then also, if the mRNA does not have this tail, it's not going to be transported out of the nucleus. So, and then obviously if you have a very short tail, so quick decay is going to happen and you could potentially lose the code, and then of course the protein is not going to be produced. So the last piece of information here as far as processing is about the intron removal. So we have to remove the introns because they do not code for the proteins. And what we have here is spliceosome complexes are going to form, and they're going to splice out, remove the introns. So the spliceosomes consist of what we call SNRPs. So SNRP is basically a protein, and this protein has a strand of RNA. And then the RNA, small nuclear RNA, is going to communicate with the consensus sequences that are in the intron so that they can recognize where exactly they need to cut. I'm going to show you a picture of what it looks like here. So this would be that SNRP, and it has that small nuclear RNA. You can see it's looped right here, and it's single-stranded, and it's complementary to the consensus sequence that's within the intron here that has to be excised. So this complex comes together, removes the intron by forming this lariat, sort of like a loop, and then splices the exons together, and therefore your mRNA molecule is going to be much, much shorter. And now, of course, we can recycle the introns by breaking them down into smaller components like nucleotides and then build another mRNA from these building blocks. So the pattern in which mRNA is being sliced in cells is going to vary. We call this phenomenon alternative splicing. So suppose you have a gene, and you can see this gene will have coding regions, non-coding regions, coding regions, non-coding regions, exons, introns. So when the primary transcript is produced, the primary transcript will have everything. So we'll have exons and we'll have introns, exons, introns. this primary transcript has to be processed. So we are removing the introns. But guess what? Not only we remove the introns, we can also remove exons. Yes. So if you look at the situation here that occurs in cell number 1, the mature RNA has exons number 1, number 2, number 4, number 5, and number 6. The exon number 3 has been removed. So if you change the sequence in the mRNA, you obviously going to change the sequence in the polypeptide because your amino acids now are going to be different. So if the amino acids in the sequence are different, the protein is going to have a different shape. So it's going to have a different shape, it's going to have different structure, so it means it's going to have a different function. And then if we look in the cell number 2, now we're missing number 2 and we're missing number 4. So you can see this protein is also going to be different. And then here's a third scenario where we spliced out number 2. So the point of this is that you have one gene that acts like a blueprint and then produces primary transcript, and different cells are going to cut that primary transcript in different ways. And therefore, the end result is going to be different proteins. So what this means, you do not have to have 1-to-1 ratio when you say you have one gene and that one gene can only produce one protein. No. This means one gene can actually give rise to many different proteins. Okay, so I've been talking about the eukaryotic transcription. I'm going to mention how transcription takes place in prokaryotes really fast. So notice we don't have a lot of transcription factors that come to the scene to initiate transcription by recruiting RNA polymerase. Instead, what we have here is a single sigma factor that is going to bind to the promoter, recruit RNA polymerase, and then the RNA polymerase is going to do its job, just like you saw in eukaryotes. You can see sigma is going to leave, and RNA polymerase is now synthesizing mRNA. You can see this little strand right here is actually mRNA. I'm going to extend that, so there you go. And then the RNA polymerase is going to run into termination sequence, and this termination sequence is going to be rich in A's and T's. So basically you're going to have a sequence that's A, followed by T, and then those A's and T's are going to be transcribed into A's and U's. And then you can see when you have A and U, they can actually form hydrogen bonds in between, and this is what happens. This tiny little hairpin-like structure is going to form, and this structure is going to signify the end of transcription. So RNA polymerase is going to dissociate from the DNA, release the DNA molecule so it can call back up again, and then basically transcription is completed. Now, one thing I want to mention before I go, as soon as we have mRNA transcript, ribosomes can actually initiate translation. So it means we don't have to remove introns because there are no introns here. We don't have to modify this mRNA anyhow. So translation of this mRNA can take place immediately while transcription is happening. So this is not the case in eukaryotes because those two processes are separate. Transcription happens in the nucleus, and then translation in the cytoplasm. This is where the ribosomes are located. Okay, so in the next video, I will discuss the next step of gene expression, which is going to be translation. I'll see you then.